Next we move to the final topic of this unit, which is specification 13. This specification asks us to describe the process of germination, including the role of enzymes. We have already discussed what germination means in relation to seeds, but now we will look at how this process actually takes place. Germination can be defined as the process by which a seed develops into a new plant when suitable environmental conditions such as water, oxygen, and an appropriate temperature are present. Germination marks the beginning of growth in a seed that eventually develops into a young plant. When the environmental conditions become favorable, the seed absorbs water and the outer covering called the testa splits open. This allows the radicle, which is the embryonic root, to emerge and grow downward into the soil. As the radicle develops, lateral roots and root hairs begin to form. The lateral roots help anchor the plant firmly in the soil, while the root hairs absorb water and mineral ions needed for growth. At the same time, the plumule, which is the embryonic shoot, begins to grow upward toward the soil surface. The cotyledons protect the plumule during this early stage of development. The energy needed for this initial growth comes from the food stored in the cotyledons. This stored food, mainly in the form of starch, is converted into soluble sugars by the action of enzymes. These sugars are then transported through the phloem to the growing parts of the seedling where they are used in respiration to release energy. The germination sequence begins when the testa splits and the radicle emerges. Root hairs soon develop to increase the absorption of water. The hypocotyl, which is the embryonic stem, begins to elongate and forms a curved hook that pushes through the soil. As the hypocotyl continues to grow, it pulls the cotyledons above the soil surface. Once above the soil, the hypocotyl straightens and the cotyledons unfold. The growing point containing the plumule then becomes exposed. Soon after, the first true leaves develop and begin to carry out photosynthesis. As the plant starts producing its own food, the cotyledons gradually shrink and eventually fall off. The process of germination can be understood more clearly when broken down into stages. First, water absorption occurs. A dry seed usually contains only a small amount of water, around 5 to 20%. When it is placed in moist soil, it absorbs water and swells. This water activates enzymes and starts the metabolic activities inside the seed. The second stage involves the activation of enzymes. When water enters the seed, enzymes in the cotyledons become active. These enzymes convert stored food substances, mainly starch, into soluble sugars. These sugars are transported to the growing regions and are used in respiration to release energy for growth. The third stage is the emergence of the radicle. The radicle is the first structure to grow out of the seed. It breaks through the seed coat and grows downward into the soil. Root hairs and lateral roots soon develop, helping the plant absorb water and minerals while also anchoring it in the soil. The fourth stage is the growth of the embryonic stem. The hypocotyl elongates and pushes upward through the soil. As it grows, it pulls the cotyledons above the surface of the soil. The fifth stage is the emergence of the plumule. The plumule, which is protected between the cotyledons, grows upward and develops into the shoot. Once it reaches the surface, the hypocotyl straightens and the first leaves begin to open. The final stage is the transition to photosynthesis. As the stored food in the cotyledons is gradually used up, they shrink and eventually fall off. The green leaves begin photosynthesis and produce food for the plant, allowing the seedling to become independent. Enzymes play a very important role in germination. When water enters the seed, enzymes become active and begin breaking down insoluble stored food such as starch into soluble sugars. These sugars are transported to the growing tissues and used in respiration to release the energy required for growth and development. Without the action of enzymes, the seed would not be able to use its stored food reserves and germination would not occur. In conclusion, germination allows a seed to grow into a new plant when the right environmental conditions are present. The purpose of germination is to start the life cycle of a new plant and ensure the continuation of the species. The benefit of this process is that it enables plants to reproduce, grow in new areas, and maintain plant populations in different environments. So, that wraps up Unit 16.3 on sexual reproduction in plants. Be sure to go through the notes linked below, and I'll see you next time. Thank you for watching.